Hello, the topic for today's microbiology class is Structure of Bacterial Cells. Bacteria are the examples of prokaryotes, and the components of bacterial cells include capsule that is present in outermost part of some bacteria, pili and flagella that is also present outside the cell wall, cell membrane, cytoplasm that contains ribosomes, nucleoid granules, plasmids, and transposons. So this picture shows the structure of bacterial cell. In the outermost part there is capsule and pili are projecting outside from the cell, and a single flagellum is shown here that is also projecting outside from the cell. After the capsule there is cell wall, then there is plasma membrane, and then cytoplasm that contains ribosomes, plasmids, and nucleoid. Nucleoid contain the genetic material of bacteria. Capsule. The capsule is a gelatinous layer covering the entire bacterium. It is composed of polysaccharide except in Anthrax bacillus, which has a capsule of polymerized D-glutamic acid. It is a determinant of virulence of many bacteria. Specific identification of bacteria can be made by using antiserum against the capsular polysaccharide that is shown by capsular swelling in a reaction called quelling reaction. Capsular polysaccharides are also used as antigens in certain vaccines, and capsule also play a role in adherence of bacteria to human tissues. Next structure of bacteria is flagella. Flagella are long whip-like appendages that move the bacteria towards nutrients and other reactants, and this process is called chemotaxis. It may play a role in the pathogenesis of motile bacteria, for example E. coli and Proteus species, by propelling the bacteria up to the urethra into the bladder and may cause UTIs. Some species of bacteria, for example Salmonella species, can be identified in the lab using specific antibodies against these flagellar proteins. Pili are also an important component of bacteria required for the motility. These are hair-like filaments that extend from the cell surface. They are shorter and straighter than flagella and are composed of subunits of pilin, a protein arranged in helical strands. They are mainly found on gram-negative bacteria. Pili have two important roles in bacteria. They mediate the attachment of bacteria to specific receptors on the human cell surface, and secondly, a special kind of pilus that is called sect pilus forms the attachment between a male and a female bacteria during conjugation. Next important component of structure of bacteria is cell wall. Cell wall is located external to cytoplasmic membrane and is composed of peptidoglycan. The structure, chemical composition, and thickness of cell wall differ in gram-positive and gram-negative bacteria. Peptidoglycan provides structural support and maintains the characteristic shape of the cell. The term peptidoglycan is derived from the peptides and the sugar that make up the molecule. It is also called murein and mucopeptide. It is composed of alternating N-acetyl muramic acid and N-acetyl glucosamine molecules, and attached to them are the transpeptide consisting of both D and L amino acids, the precise composition of which differs from one bacterium to another. Peptidoglycan is basically a good target for antibacterial drugs. Several of these drugs such as penicillin, cephalosporins and vancomycin inhibit the synthesis of peptidoglycan by inhibiting the transpeptidase that make the cross-link between the two adjacent peptides. This table shows the difference between the cell walls of gram-positive and gram-negative bacteria. Peptidoglycan in gram-positive bacteria are thicker and multi-layered, while in gram-negative they are thinner and single-layer. Teichoic acid is present in the cell walls of gram-positive bacteria while not present in gram-negative bacteria. Lipopolysaccharide or endotoxin is present in the cell wall of gram-negative bacteria. This picture shows the difference between the cell walls of gram-positive and gram-negative bacteria. In gram-positive bacteria, there is thicker peptidoglycan layer, while in gram-negative the peptidoglycan is thinner and single layer. Teichoic acid is present in the cell wall of gram-positive bacteria, while lipopolysaccharide is present in the cell wall of gram-negative bacteria. There is also an important space called periplasmic space that is present between the peptidoglycan and the inner membrane. This space is important because it contains a species of enzymes called beta-lactamase that are important for the degradation of various antibiotics including penicillin, cephalosporins and causes their resistance. Cell wall of acid-fast bacilli. Mycobacteria species, for example Mycobacterium tuberculosis, have unusual cell wall that result in their inability to be gram-stained. These bacteria are called acid-fast because they resist decolorization with acid alcohol after being stained with carbamazepine dye during the procedure of gram staining, and this property is due to the high concentration of lipid called mycolic acid in their cell walls. This picture shows the cell wall of mycobacteria that shows the high amount of mycolic acid in their cell wall that resists their ability to decolorize with the acid alcohol. Next important component is cytoplasmic membrane. Just inside the peptidoglycan layer of cell wall lies the cytoplasmic membrane that is composed of phospholipid bilayer. They are chemically like eukaryotic membrane, but eukaryotic membranes contain sterols while prokaryotes do not contain sterol. The only prokaryotes that have sterols in their membrane are members of genus Mycoplasma. Important functions of cytoplasmic membrane include active transport of molecules into the cell, energy generation by oxidative phosphorylation, synthesis of precursors of cell wall, and secretion of enzymes and toxins. Next component is cytoplasm that has two distinct areas when seen under electron microscope. First is an amorphous matrix that contains ribosomes, nutrient granules, metabolites and plasmids, and second is inner nucleoid region composed of DNA. Ribosomes. Bacterial ribosomes are the site of protein synthesis as in eukaryotic cells, but they differ from eukaryotic cell because they are 70S in size with 50S and 30S subunits, while eukaryotic ribosomes are 80S in size while with 60S and 40S subunits. Next component is granules. The cytoplasm contains several different types of granules that serve as storage areas for nutrients and they stain with certain dyes. For example, metachromatic granules are characteristic feature of Corynebacterium diphtheria that is the cause of diphtheria as shown in this picture. Next component is nucleoid. That is the area of cytoplasm in which DNA is located. DNA of prokaryotes is single circular molecule. Bacterial DNA has no introns, no nuclear membrane, no nucleolus, no mitotic spindle, and no histones, while the DNA of eukaryotic cell contains all these structures. Plasmids. Plasmids are extra-chromosomal double-stranded circular DNA molecules that can replicate independently of bacterial chromosomes. Several different types of plasmids can exist in one cell. For example, transmissible plasmids can be transferred from cell to cell by conjugation, and they are responsible for the synthesis of Sec pilus and for the enzymes required for the transfer. While non-transmissible plasmids are small since they do not contain the transfer genes, they are frequently present in many copies per cell. Plasmids carry the genes for the following functions and structure of medical importance. First is antibiotic resistance, that is mediated by a variety of enzymes such as beta-lactamase. Next is exotoxins, for example enterotoxin of E. coli, anthrax toxin of Bacillus, Pili or Fimbria, which mediate the adherence of bacteria to epithelial cells, resistance to heavy metals, for example mercury and silver, that is mediated by reductase enzyme, resistance to UV light, that is mediated by DNA repair enzymes. Transposons. Transposons are pieces of DNA that move readily from one side to another, either within or between the DNAs of bacteria, plasmids, and bacteriophages. Because of their unusual ability to move, they are also called jumping genes. Some transposons move by replication, replicating their DNA and inserting the new copy into another site, that is called replicative transposition, while others are excised from the site without replicating and then inserted into another site, that is called direct transposition. Transposons have four identifiable domains. On each end there is a short DNA sequence of inverted repeats which are involved in the integration of transposon into the recipient DNA. The second domain is the gene for transposase, which is the enzyme that mediates the excision and integration process. The third region is the gene for repressor that regulates the synthesis of both the transposase and the protein encoded by the fourth domain, which in many cases is an enzyme mediating antibiotic resistance.